Hello everyone. Today we are going to learn how to effectively review and answer multiple choice questions or MCQ from past year exam papers. This will help you prepare better and improve your exam performance. Reviewing past papers is important because it helps you understand the exam format, identify common topics, and practice time management. And it's also highlight your strength and area for improvement. The photomicrograph show the atomic structure of part of a cell, which is statement about the type of cell shown in the photomicrograph is correct. The answer will be B. Which structures are found in typical eukaryotic cells? In typical eukaryotic cells, you would find ATS ribosome and also linear DNA chromosome. 470 ribosomes and circular DNA are typically found in prokaryotic cells, such as bacteria, although 70S ribosome and circular DNA can also be found in mitochondria and chloroplast of eukaryotic cells. A student examined a slide of human blood with a light microscope and made a careful drawing of the different cell types. The student used an eyepiece reticule so that the relative sizes of the different cell types were drawn accurately. What is the linear magnifications of the drawing? To calculate the linear magnifications of the drawing, you need to compare sizes of the cells in the drawing with the actual sizes. The average diameter of the red blood cells is approximately 7 micrometers. The diameters in the drawing is given as 7 millimeter. And first, what you need to do is you need to convert the millimeter to micrometer. 7 mm is equivalent to 7,000 micrometer. And now, to calculate the magnifications, you have to use the formula which is size in drawing divided by the actual size. With 7 micrometer divided by 7,000 micrometer divided by 7, you get the value of 1,000. So the answer will be X or multiply 1,000. The diagram shows a graduated slide with divisions of 0.1 mm view using an eyepiece reticule. Pollen grains were grown in a sugar solution and viewed using the eyepiece reticule. Diagram 1 shows the pollen grain at the start. Diagram 2 shows the pollen grains after 4 hours. What is the growth rate of the pollen tubes? The answer will be 5. The table shows a comparison between two pictures of a typical eukaryotic cells and the typical prokaryotic cells. Which row show the correct comparison between these cells? Let's look next page for further explanation. To determine the correct comparison, let's review the features. For centromeres, eukaryotic cells have centromeres, which are regions of chromosome where the cytocross form the spindle fiber attached during cell division. Prokaryotic cells do not have centromeres because they typically have a single circular chromosome that does not require centromeres for cell division during cell division. But for telomeres, eukaryotic cells have telomeres, and which are repetitive nucleotide sequence at the end of linear chromosome that protect the end from deteriorations or fusions with neighboring chromosome. Prokaryotic cells do not have telomeres because their chromosomes are usually circular and do not have ends that require protection. So the answer will be B. The flow diagram shows the result of a number of tests on a solution of biochemicals. Benedict test turn it to be blue is indicate that no reducing sugar. For the non-sugar test is indicate in yellow color. This is actually the signs for non-reducing sugar present. Example, sucrose. And iodine test, blue-black. This is also portrayed that is a starch present, and blue-red test it turned out to be purple, which indicate protein present. So the answer would be sucrose, starch, and catalase. Maltose and sucrose are disaccharides. Maltose is formed from two molecules of glucose, where sucrose is formed from fructose and glucose, which will show the molecular formula of the two disaccharides. The molecular formula for both maltose and sucrose is the same, C12H22O11. This is because both are formed by the condensation of two monosaccharides, glucose for maltose and glucose fructose for sucrose, with the loss of one molecule of water. So the correct answer will be A. The diagram shows short section of some common polysaccharide and modified polysaccharide, and which row correctly matches the number diagram to the description of the polysaccharide. The answer will be B. Number 9, which molecule contain at least two double bonds? Unsaturated fatty acid contain at least two double bonds. Collagen does not contain double bond, and hemoglobin contains double bond in its table. So the answer will be 1 and 3. Number 10, the diagram shows a protein molecule as you see in the diagram, and two long polypeptides each form alpha helices for much of their length, and these twist together into a fiber. At one end, each of these polypeptides coil into a globular head. Two short polypeptides bind to each globular head. And what actually describes the protein structure of this molecule? The answer will be a quaternary structure because each molecule consists of 6 polypeptides. 11. Which type of bond will keep a folded protein in its precise shape for the longest time as the temperature rises? The answer will be disulfide. Why disulfide? Disulfide bond are covalent bond formed between two sulfide within residue within a protein. And these bonds are quite strong and stable, which help them maintain the folded structure of protein over a longer period. Even as temperature increases, disulfide bonds are particularly important in stabilizing the tertiary and quaternary structures of protein, contributing significantly to their overall stability. Protease enzymes are found in many locations inside and outside the cells. 4 of these locations are listed: lysosom and at the telophase spindle. These locations are sites of intracellular hydrolysis. An enzyme is modified for industrial use. It has a lower Michael-Menten constant than the unmodified enzyme. What is a true of the modified enzyme? A lower Km means the enzyme binds the substrate more tightly, which is indicate higher affinity. So the answer will be B. 14. A decrease in some factors will increase the fluidity of the cell's membrane. Which pair of factors, when decreased, will increase the fluidity of the cell surface membrane? The answer will be a decrease in proportion of phospholipid with saturated fatty acid change, and a decrease in the proportion of a long fatty acid change. The diagram shows a cell surface membrane, which a statement about the label molecule in the membranes are correct. The answer will be 2 is involved in facilitated diffusion, and 4 is involved in membrane fluidity. Equal-sized potato peas were placed into test tube containing equal volume of different concentration of sucrose solution and left for 30 minutes. All other variables are controlled. After 30 minutes, the potato peas in one of the concentration of sucrose solution had not changed in size. What can be concluded from this result? There is no net movement of water into or out of the potato, and the water potential of the potato is the same as the water potential of the sucrose solution. Which features of the stem cell enable them to replace cells in tissues such as skin? Stem cells can replace cells in tissue like the skin because they divide by mitosis to supply some cells that can differentiate. During prophase, a scientist stain the chromosome of a diploid animal cells with a fluorescence dye to stain telomeres. And these cells has 32 chromosomes. And how many telomeres were the scientists observed? Each chromosome has two telomeres, one at each end. In a different animal cells with 32 chromosomes, 32 chromosomes multiplied by 2 telomeres per chromosome, we get the value of 64. Number 19, the diagram shows the cell cycle, during which phase two chromosomes condense and become visible. So it should be at the phase of D. What is the correct for timing? The answer will be D. Has a single structure, has is a pyrimidine, and incorrect for join in its complementary base with 3 hydrogen bond. 21. A short piece of DNA 15 base pair long was analyzed to find the number of nucleotide bases in each of the polynucleotide strand, and some of the results are shown below. How many nucleotides containing guanine were present in the strand? The answer will be 2. Scientists grew bacteria in a medium containing heavy nitrogen as the only source of nitrogen. And after many generations, all of the bacteria that contain heavy nitrogen. And these bacteria were then moved from the heavy nitrogen medium into a medium with only light nitrogen. And some bacteria were collected from each of the next 3 generations, and their DNA was analyzed. Hybrid DNA contain both heavy and light nitrogen, and which row show the correct DNA of the first and third generation? The answer will be B. A population of bacteria is exposed to the antibiotic penicillin, and most of the bacteria die. However, some bacteria in the population have an allele coding for an enzyme that breaks down penicillin, and these bacteria are able to survive, which could explain how these bacteria cells acquire this allele. So the answer is 1 only, because an error during DNA replication can introduce mutation, and including the one that could result in allele coding for an enzyme that breaks down penicillin. An error during transcription and translation do not introduce permanent changes to the DNA sequence, and thus cannot account for the presence of a new allele in the bacterial population. Question number 24. A dicotyledonous leaf has a palisade mesophyllalia that is approximately twice as thick as spongy mesophyllalia. Which plant diagram is correct? The answer will be C. A number of processes contribute to maintain a water potential gradient in plants, allowing water to reach the highest part of a plant. And which processes are responsible for maintaining this water potential gradient? The answer will be during the process of osmosis and transpiration. Osmosis allows water to move from area of high water potential to the lower water potentials within the plant cells. While transpiration, the evaporation of water from plant's leaf, create a pool that draws water upward through the plant. Capillary also help, but not a major factors in maintaining the overall water potential gradient in tall plant. And what is the correct statement about the movement of substances absorbed by roots? The correct statement about the movement of substance absorbed by root is soil water have a more negative water potential than the xylem cell. This statement correctly reflects the process of water and mineral ion uptake by the plant root. Water and ion move from the soil into the root cells, where they are transported through the xylem to the rest of the plant. 27. Which arrangement of 4 molecules of water show how water may cohere when moving up a xylem vessel? The answer will be B. And which statement about sucrose loading into companion cells and then into a polyunsaturated element is not correct? The statement that is not correct is sucrose molecule move from the companion cells into the seed tube of the folium tube plus modesmata. Plus modesmata are channels that connect plant cells, which allow for communication and transport of substances, but they do not play a direct role in the transport of sucrose from companion cells into seed tube. Instead, sucrose moves from companion cells into seed tube via specialized transport mechanism, which involve membrane protein and active transport processes. So the answer will be D. And number 29, what explain why the left and right sides of the heart contract simultaneously? The answer will be there is no barrier to electrical excitation between two sides of the heart. And this is because the electrical impulses generated by the sinoatrial or SA node can spread freely across both atria and then through the ventricular AV nodes to the ventricle, allowing for synchronized contractions on both sides of the heart. And number 30, the statements leave some of the events in the cardiac cycle. They are not in the correct order. Which statement describes the third of this event to occur in the cardiac cycle? This is a common question which already exists in May 2021. So, the third event in the cardiac cycle based on the correct order is atrioventricular nodes delay the impulse for fractions of a second. And the sequence of event would be, first step is the sinoatrial nodes contract. Step 2, a wave of excitation sweep across the atria. Step 3, the atria contract. Step 4, the atrioventricular nodes delay the impulse for a fractions of a second. Step 5, the impulse travels through the Purkinje tissue. And then step 6, the wave of excitation sweep outward from the base of the ventricles. And finally, step 7, the ventricles contract. The diagram shows pressure changes in different parts of the heart during one cardiac cycle, during which periods the semilunar valve open and the atrioventricular valve close. The answer will be C. 32, which diagram show the event that occur during transport of carbon dioxide by the blood? So for this question, the answer should be C. And number 33, which structures are found in bronchi? We have cartilage, we have smooth muscle, and we have goblet cells. Smooth muscles is used to help in the contractions and dilation of the bronchi. Cartilage is used to provide structural support to keep the bronchi open, and the goblet cells produce mucus to trap gas and microorganisms. So the answer will be B, which is structures that found in bronchi would be smooth muscle, cartilage, and goblet cells. Number 34, which tissue in the respiratory system is correct linked to its function? The correct will be cartilage keeps airway open, ciliated epithelium remove particles from the air, and the elastic fiber head lung stretches and recoil, and the smooth muscles adjust the diameter of bronchioles. So the answer will be B, which is ciliated epithelium which remove particles from the air. And chronic obstructive pulmonary disease, or so-called COPD, includes emphysema. Which effect does emphysema have on gases exchange? So the correct will be surface area to volume ratio of lung decreases. Emphysema destroy alveolar walls, reducing the surface area available for the gas exchange. And the third one will be volume of oxygen diffused per unit time decreases. With less surface area, less oxygen can be absorbed into the bloodstream. Number 36. The diagram shows some of the pathogens that cause disease improvement and some of the way they are transmitted, and which row matches the correct number of the pathogen with the correct letter for the modes of transmission for cholera and measles. That will be the answer for A. And 37, which factors could increase the global distribution of malaria? The answer will be 3 and 4, a rise in global air temperature, the increasing irrigation of land for farming. A rise in global of air temperature is a warmer temperature expand the habitat of mosquito that transmit malaria. And number 4, the increasing irrigation of land for farming, which create more standing water, which is a breeding ground for mosquitoes. 38, the antibiotic Teixobactin was discovered in January 2015. Teixobactin kills some bacteria such as Staphylococcus and Mycobacterium, and most antibiotics work by binding to protein. Teixobactin binds to lipid that are used in the synthesis of bacterial cells wall. This means that it is unlikely that bacteria will quickly develop resistance to Teixobactin, and which statement explains why bacteria are likely to quickly develop resistance to Teixobactin. That will be a mutation in the gene coding for a protein allows selections for resistance. And bacterial resistance often arises from mutation in gene encoding protein, which then gets selected for. And then the structure of a lipid is not encoded by DNA. Since Teixobactin targets lipid and lipid structures are not directly encoded by DNA, bacteria cannot easily develop resistance through genetic mutation. And number 39, which row correctly describe the type of immunity gain from being injected with the antitoxin? So the correct answer will be C, where active immunity gain when the body immune system responds to a pathogen or vaccines, and passive immunity gain when antibodies are directly given, such as through antitoxin. And artificial immunity is acquired through medical intervention like injection, and the natural immunity acquired through natural exposure to the pathogen. So the answer will be C. And 40, there are antibodies found during an immune response. The correct will be A. On surface of pathogen, antibody binds to pathogen to neutralize them. And in blood plasma, antibodies circulate in the body, in the blood to find and bind to pathogen. That's all for today's presentation. And thank you very much for your support and cooperations. And through regularly practicing with the past paper and using these strategies, you will become more confident and efficient in answering MCQ. Good luck with your studies and upcoming examination. And don't forget to subscribe to John Study Lab YouTube channel.